Today's lecture is going to be on general senses with an emphasis on vision, and I'm going to do a little bit of an introduction on hearing so you can learn about some of the anatomy. I'm not going to go into great depth on hearing, but we'll touch on it a little bit. So let's do an introduction, as we always do, and talk about, so we're coming off of the nervous system lectures, and we're going to kind of tie all of this together. Let's talk about some of the receptors that are in our body too. So our bodies are constantly exposed to sensory information, which is called stimuli. And our conscious awareness to these stimuli is called sensation. And that's a topic we've touched on throughout the nervous system lectures. And all of these different types of stimuli are detected by receptors that are throughout our entire body. And these receptors are broken up into categories. So there's something called general senses, and I'm going to touch on the different types of receptors on the next slides. And these receptors are monitoring things like temperature, pain, touch, stretch, and pressure. And then we have other type of senses called special senses. And this includes gustation, which is the sense of taste, olfaction, which is the sense of smell, vision, equilibrium, and auditory. So that is our hearing and our balance. So I'm going to give some examples, a very short example on gustation, but I'm mostly going to focus on vision and then give a little bit of an introduction to equilibrium and hearing. So this slide represents and talks about the different types of receptors that are monitoring all of these different sensations. So let's just go over these receptors and get a general idea of what they're doing. So the first type of receptor that I want to talk about are chemoreceptors. And these can be found on the tongue. So these are these receptors respond to changes in chemical concentrations. You can also find these types of receptors throughout the body to monitor some of the pH changes that occur. We also have pain receptors. These can respond to tissue damage. They're also called nociceptors. And then we have thermoreceptors, which, as the name implies, responds to changes in temperature. We have mechanoreceptor, which respond to mechanical forces, and this can be found in areas like joints or muscles. Photoreceptors are found in the eye. They respond to light. And then we have osmoreceptors. These can respond to changes in solute concentration, so salts and ions and things like that. And then finally, we have something called baroreceptors, which can respond to and detect changes in pressure. So you can find these in some of the larger vessels of the heart and even in the atrium of the heart, where changes of pressure are going to be important for normal heart activity. All right, so all these receptors can respond to stimuli in two different ways. So we have something called tonic receptors and phasic receptors. So let's talk about phasic receptors, and I also have another slide that shows this in more of a graph format. So phasic receptors, we have a rapid adaptation to it. So initially, there may be firing of that receptor to transmit that signal, but over time, we adapt to it. And this is to ignore unimportant information. So it's just like, a smell that you might get, like a really strong sensation of something, and then over time it kind of, you get used to it. You stop smelling that, whatever noxious smell it might be. This can also be like when you originally put your socks and shoes on, you'll feel that sensation on your feet. And then over time, as the day goes on, that information stops being transmitted. Whereas tonic receptors, this is the opposite. They have little to no adaptation. So this is going to be a continuous signal for the entire time that the stimulus is there. And there are types of receptors where we don't want that signal to go away. And an example would be pain. So this is something that is being monitored continually and must be continually evaluated. So let's take a closer look at both tonic and phasic receptors. So again, here's those phasic receptors where the stimulus is applied, but over time we get used to that stimulus. So we're adapting to it. Whereas the tonic receptors, the action potential, and we really didn't talk about how neurons transmit their signals from these receptors. So I'm not going to go into a whole lot of detail because that's more physiology, but you get the idea that the signal in a tonic receptor is maintained over time. So again, with phasic receptors, after a while, you no longer sense the effect. Whereas tonic receptors, that signal is going to be constant. You're never going to adapt to it. So an example of that would be nociceptors. So we don't want to adapt to that pain. We want to, the brain is being or constantly being notified of that signal. So we've spent a lot of time talking about how nerves transmit their signals. We talked about the spinal cord, and we talked about the brain and how the axons and neurons are coming together. And this is kind of putting everything in a big picture. And this is just one small example of how these receptors are working. And a good example is to think about taste buds. And these are the receptors for taste. So all along the tongue, so here is the tongue, and then you have a blown up image of it showing you these taste buds, which are the receptors to that are aligning with these nerves. So these are the sensory nerves right here that are in connection to these taste bud receptors, so that the signal can be transmitted to the brain. So each taste bud is associated with a sensory nerve fiber. And there are various locations on the tongue that have the ability to sense different types of foods or flavors. So what is happening, and again, we've kind of talked about this, but this is putting it all together, is that you have the receptors on the tongue, you have the nerves that are associated with those receptors. So when a sensation is detected, that information is transferred along the nerve, and we've talked about some of these cranial nerves that actually innervate the tongue. That signal can then be transferred to the medulla oblongata, filtered and relayed, and then sent to specific areas of the cerebrum to interpret that taste. So this is an example of a typical pathway where you have these sensory receptors associated with nerves, and those nerves then communicate back to the brain for interpretation. All right, so that was one example. I want to take some time and talk to you about vision, and I want to go through the anatomy of the eye, and then we'll talk about the functional regions of the eye. So the eye is the visual organ. It is where we are going to determine and process all of the information that we are seeing. So we've talked about sensory receptors, and it turns out that 70% of all the sensory receptors that are in our body are located in the eyes. There's a tremendous amount of information that is being processed at any given time from the eyes. So a lot of the cerebral cortex is actually involved in processing all of this visual information. And what I'm going to go on to show you, and we're going to talk about the structure of the eye, is that that there are layers to the eye. And these layers are called tunics. And that's actually how I have broken down my lecture. We're going to talk about the different tunics of the eye. Before we talk about the structure, though, I want to just go over some of the external accessory structures that help keep the eyes clean and prevent them from infection or any kind of damage that could occur. Most of these structures on this slide, it's relatively intuitive as to what their jobs are, but we'll go through them anyway. So of course we have eyebrows, which help prevent sweat from dripping into the eye. We have eyelashes that prevent large foreign objects from contacting our eyes. Our eyelids, which are movable, we learned about some of the muscles around the eye, provide a protective covering for the eye. And then there are some glands that are associated with the eye that we'll talk about. One of them is called the tarsal glands. They are located within both eyelids, and they contain sebaceous glands. And we talked about sebaceous glands when we were working with skin. And these help to prevent the eyelids from sticking together. It also keeps the eyes moist. All right, let's talk about a couple other structures before we get into the tunics of the eye. And one of them is called the conjunctiva. The conjunctiva lines the insides of the eyelids and covers the sclera. The sclera is the white part of the eye. The conjunctiva is made up of stratified squamous epithelial cells, as well as goblet cells. Goblet cells secrete mucus. If you remember from some of our histology slides when we were looking at intestine, you probably saw goblet cells. And so this is a transparent mucous membrane that kind of sits on the surface of the eye. And what this conjunctiva is doing is providing protection and lubrication of the eye by producing mucus and tears. So we also have other glands that are going to secrete tears. So there is a lacrimal gland, which I'm going to talk about in a minute, that produces tears. But so in combination with the lacrimal gland, the conjunctiva does provide lubrication to the eyes. And you've probably heard of conjunctivitis, right? So that is when we get an infection of this epithelial lining, and it sometimes causes your eyes to turn red and cloudy, and that's usually easily cleared up with an antibiotic drops. All right, so let's turn our attention to the production of tears and how the lubrication of the eyes occurs. So there's a very sophisticated apparatus called the lacrimal apparatus. that is going to produce tears. And there's several structures that are associated with this. So again, the function of the lacrimal apparatus is to keep this anterior surface of the eye moist. And these tears help to prevent bacterial infections. They contain an antibiotic-like enzyme called lysozyme. And lysozyme helps to lyse bacteria, hence its name. And it can help keep bacteria away from our eyes. So the way that this apparatus works is, and this is a really great picture here, it's showing you the lacrimal gland here, okay? This gland is going to produce lacrimal fluid or tears, and these tears will flow across the eye. On the medial side, there is, and I've kind of circled some of the structures that I want you to know. So on the medial side of the eye, there are what's called lacrimal canaliculi. And you're now familiar with the term canaliculi. They're little canals that will then absorb some of these tears as they are shed. And those lacrimal canaliculi will lead to another structure called a lacrimal sac. This lacrimal sac will then drain through the lacrimal duct into the nasal cavity. And that's, you kind of can think of this as that's why your nose will start running if you're crying pretty heavily, because you're getting all of this drainage into the lacrimal sac down through the nasal lacrimal duct and into the nasal cavity. So I'm going to just highlight these terms that I want you to know. There are a couple others that I didn't go over, so I just wanted to point out the ones that I did. That's all that you ever need to know. I believe we have a model of this in the lab too, so you can work with the model and see the different structures that make up the lacrimal apparatus. All right, moving on. Now we're going to get into the anatomy of the eye. So let's talk first about the chambers. So there are two chambers within the eye, and they're both under pressure. We have something called an anterior chamber and a posterior chamber. Why do I not see the posterior chamber? Anyway, I'll just point to it. It's not labeled. So the posterior segment, that's the larger cavity. It is filled with something called vitreous humor. This is a clear, jelly-like substance, and when we do the dissection of the eye, you'll actually get to see this vitreous humor. And this is going to transmit light. There are going to be receptors all here on the back of the eye, on the retina, that are going to absorb light. This vitreous humor also supports the posterior surface of the retina. So because it's under pressure, it's going to push the retina back against the eye so that there are no wrinkles in this, right? We don't want any wrinkles. Sorry, my tracing's not great. And so that fluid is going to push that retina up against the back wall. The anterior chamber is filled with a different type of solution called the aqueous humor. And this is much thinner. It's more like water. It is renewed continuously, and it supplies nutrients to the anterior structures, such as the lens, which is right here, and the cornea, which is the surface of the eye. So those are the chambers of the eye. And again, they're both under pressure. You may remember going to the eye doctor, or the optometrist or an ophthalmologist, where you put your face in this contraption and they blow air at your eye. This is to determine that you have the right amount of pressure in your eye. All right, now we're moving on to the tunics of the eye. So there are 3 tunics of the eye, 3 layers that we're going to talk about. And the way I've organized this lecture is to go through and talk about each of these tunics in order. So I'm going to start with the outer tunic, which is called the fibrous tunic. We'll talk about the structures that make up the fibrous tunic, which is the sclera, the white part of the eye, and the cornea. We'll then move on to the vascular tunic, where we'll talk about the iris, the ciliary body, and the choroid. It's a blood supply that is needed for the eye. And then we'll talk about this inner layer, which consists of the retina. And this is where all those vision receptors are, all right? And we'll talk about how the nerve, also at the back of the eye here, is part of this layer. All right, so we're going to talk about the fibrous layer first. All right, so this is a close-up. You can see here's the structure of the eye. We're going to talk about the sclera first. That is the white portion of the eye. It's not just the visual part that you see in the front. It actually extends all around the eye. So you can see it here, all around the eye here. And the sclera, it has an important role in that it is the site, the attachment site for the eye muscles, which we will get to later on in the lecture. And we'll talk about the different muscles that attach to the eye to allow us to move our eyes in different directions. The sclera is made up of 3 components. There's 3 layers to the sclera. It's actually pretty tough, and you'll see this when we do the dissection. So the first part of the sclera is called the episclera. This superficial region is going to provide nutrients to the eye. And then there is a stroma, a middle layer. This is what's giving it its structure. There's a lot of collagen fibers that are running through the stroma layer, giving the sclera its tough appearance. And then finally, there's the lamina fusca. There's going to be blood vessels that run through the lamina fusca, and this is going to provide oxygen to the eye. So through exchange of oxygen so blood carries hemoglobin, which also carries oxygen. We'll talk about that when we get to blood. But that is how those exchanges of gases occur. It's going to always happen at the blood vessels. So that's the sclera. And then the other structure that is associated with this tunic is the cornea. And the cornea is this outer region right here. It's a transparent, dome-like structure that is going to cover a lot of the internal structures of the eye and protect the eye. So what is the cornea doing? So the functions of the cornea are that it's going to perform almost 70% of all of the focusing that we do with our eyes. Again, I've talked about how it is the outer layer, so it's going to protect the eye from the outside world. The cornea does not have a blood supply, so it is cleaned and nourished by the tears and the aqueous humor that's in that anterior chamber that we talked about. And this, the cornea also contains pain receptors because it's very important for us to be able to detect if something is in our eye, such as an eyelash, so that we can remove it so that it doesn't cause damage. Now we're going to move on to the next tunic. So we just covered the fibrous tunic. We talked about the sclera and the cornea. And now we're going to move on to the vascular tunic and talk about these structures that you find here. So we're going to talk about the iris, which, as you know, is the colored part of our eye. And then we're going to talk about the ciliary body and the choroid. But let's talk about the iris first. So again, this is the visible colored part of the eye. This determines our eye color. And as you know, it can be brown, green, blue, hazel, any of these different types of colors that you see. This can change based on genetics. It is composed of smooth muscle, okay? Remember that we don't control smooth muscle, right? It is under the control of the nervous system and the autonomic nervous system. And this is what is going to decide and control the size of the pupil. So you can see the iris here in this picture. And now this is a nicer view, so you can really see how that ring is going to, like the size of the circumference of the pupil is going to change. So here is the iris here. And there are muscles that are associated with the iris. And this can change the opening or the size of the pupil. So the pupil here is in the middle. This is that round opening. And depending on what, if we're in a sympathetic nervous system state or a parasympathetic, remember we sometimes reside somewhere in the middle, but the sympathetic nervous system is your fight or flight, right? Fight or flight. And in this situation, we want to be able to see everything around us so that we can determine how to react. So we're going to be quite dilated. So there is a muscle that is innervated, because remember the autonomic nervous system, there's innervation of both systems, both the parasympathetic and the sympathetic division are innervated by two different neurons, one for each pathway. So there's going to be a dilator pupillae muscle for the sympathetic nervous system, and then there's going to be a sphincter pupillae muscle for the parasympathetic nervous system. And remember, each are controlled by the different divisions depending on what state you are in. So that's going to determine, it's going to decide the size of the pupil. So if you're in rest and digest, if you have no stress and you're just chilling out, we kind of talked about that, the sphincter pupillae muscle is going to constrict because you don't need a lot of light entering your eye. So that is how the iris is controlled. And just to bring kind of a very basic picture, when you are dilated, you have an enormous amount of light coming into the eye through the pupil, and then the retina, again, this is where all those photoreceptors are so that we can determine what it is we are seeing. The visual field is much greater. When you have an undilated pupil, you have a lot less light coming in and a lot less information to be processed. The other structure is the lens. All right, and you'll see this when we do the dissection too. I kind of describe it as two contacts put together is what it kind of looks like. So it's a relatively thick but very transparent biconvex disc. Okay, so it kind of looks like that. It's not the best picture. And it sits behind the iris. And what its function is to refract the light that is going to be focused on the retina, okay? And there is something called a ciliary body that is going to be able to change the shape of the lens. And I'll show you that on the next slide. And that's going to help refract that light. So it's going to help our eyes focus objects at various distances. Let me show you an image of the ciliary body. So again, here's an image of the eye. You can see the lens right here. Here is that ciliary body, and you can see that it is connected to the lens. And with these filaments, it will allow the lens to change its size or shape so that light can be passed back onto the retina. There are ligaments right here called suspensory ligaments that are attached to this ciliary body. The ciliary body is actually a muscle that will be able to contract and change the shape of the lens. This muscle is referred to as the ciliaris muscle. And again, as light is entering here, depending on what we're trying to focus on, the shape of the lens will change so that the right amount of light will be refracted to the back of the eye. It's also important to note, so we're talking about this layer, which is the vascular tunic. And so most of this vascular tunic is composed of something called the choroid. And the choroid, this whole layer that you see in light pink here, this whole layer is a vascular layer. There are a lot of blood vessels. My drawing is horrible. There are a lot of blood vessels that are in this area to help exchange gases. So that is what this layer is made up of. So I'm just going to highlight some of these terms for you. So the vascular tunic, again, is made up of the iris, the ciliary body, and the choroid layer. And now we're going to move on to that third layer, which is the retina. This is the innermost layer of the eye. This is where those receptors will be. Those light receptors will all be on the retina to be able to absorb light. So those photoreceptors are actually neurons. They're receptors that have axons, neurons associated with them to transmit that information back to the brain. So you've probably seen this image if you have been to the eye doctor, which I'm sure most of you have. This is a nice image showing you what the retina looks like. And I want to talk about the different structures that you'll find in the retina. It's kind of a complicated area that has a lot of different structures here. So let's work through the different structures that are in the retina and talk about what the retina is doing. So again, it is a thin layer of nervous tissue that lines the back of the eye. And so when light enters the eye, it's going to pass through the iris to get to the retina. And the light is going to pass through the lens as well. So images are focused and converted to electrical impulses because that's what the nerves are going to sense. And they're going to be carried, so there's the optic nerve that's going to reside here at the back of the eye. And this is what's going to transmit all of that information to the brain. All right, so there's a couple really important structures on the retina. So the optic nerve, so that's what you're seeing right here, is in this posterior segment of the eye. You're going to find the optic nerve. And that is where all of the axons that are going to be transmitting the information detected by the retina back to the brain. So you can find the optic nerve. There's always around the whole nerve, because it's a big cluster of axons, there is going to be an optic disc. This is the point of entry for the optic nerve. And it's actually considered to be a blind spot because there are no receptors at this point. So there's no receptors anywhere around this optic disc. There are photoreceptors everywhere else though. So there are lots and lots of axons leaving this leaving the eye at this particular spot. And just to give you some reference, there is about 1 million afferent nerve fibers from the eye to the brain. So that's all that information that's being detected is being transmitted by these nerves. So you can also see this choroid layer because you can see all the vasculature of the eye. And so again, there's photoreceptors that are all along the retina. But there's also 2 other really important spots. So there's what's called the macula luteae, and this is an oval-shaped, darkened area of the retina. And this area is responsible for the highest resolution that is possible in bright light. So there's going to be, if you remember the receptors, the photoreceptors for the eye, they're called cones and rods. So this is where you can get the highest resolution of bright light. And then there's another little spot within the macula luteae called the fovea centralis. That's what this little area right here is. The fovea centralis has the highest concentrations of cones. And again, you're going to learn more about cones and rods and physiology, so I'm just kind of giving you the basics here. But cones are responsible for having really sharp vision, which is what we need when we're reading. So both the fovea centralis and the macula luteae are really important because they have areas that have the highest resolution in bright light. This next slide is really just a summary slide of everything that I've gone over about the retina. So again, the macula luteae has the majority of the receptors kind of all clustered in that region. And then the fovea centralis has a very dense population of cone receptors, which is really important for reading, and it is located within the macula luteae. So don't get those 2 confused. And then we also have that blind spot, which is where the optic nerve resides. All of those axons together produce an optic disc, and there's no receptors or ability to see in that region. All right, and this is just, again, I didn't want to go too much into the details of the types of receptors. There are photoreceptors, that's what this slide is showing you here, all along the retina. And these photoreceptors are what is going to convert light into visual information. So everything that we're seeing, it's going to convert that information so that our brains can interpret what it is we're seeing. We have 2 types of photoreceptors, rod cells and cone cells. They kind of act in opposite ways, where rod cells are more sensitive to light and allow vision in dim light, whereas cone cells operate best in bright light. This is also where we get our color vision from. And then finally, we'll talk about the types of muscles, as I was mentioning, that allow the eye to move. We have great models of these eye muscles within the lab. So there are several of them. We have what's called the superior rectus muscle right here. That is what is going to allow the eye to move upward. We have an inferior rectus muscle. You can get downward movement. And then you're going to get medial and lateral movement. So there's a medial rectus, so it's properly named. So your eyes can look inward towards your nose. And then you have the lateral rectus for outward movement. There is one other muscle called the inferior oblique. This allows our eyes to move sort of at an angle. This is another representation so you can see all of those muscles. And again, I would urge you to look at them on a model as well so you can see them in 3D. I have one other image that is also summarizing these extrinsic eye muscles. There's no new information on this. It's just merely giving you another view so that you can see this inferior oblique muscle that kind of resides in the posterior location. Now that we've talked about the basic anatomy of the eye, we can go on to discuss some of the disorders or diseases that occur within the eyes. And I really like these images because it shows you what a person what their vision might look like if they had these particular disorders. So this first one right here is called macular degeneration. And we just spoke about the macula lutea and the fovea centralis that is located within it, and how there are a high density of photoreceptors in that area. So you can have something called macular degeneration, where that area on the retina starts to degenerate and you start to lose vision. So what happens is a lot of these photoreceptors get damaged. This is often age-related. No one knows exactly what causes macular degeneration, but they think that it could be genetics or environmental factors. And you can see what that vision would start to look like here in this image. The other disease is called glaucoma. And this is a nice view of what your field of view would look like if you had glaucoma. So we've talked about the importance of nerves, and so that optic nerve that's in the posterior region of the eye starts to get damaged. So the information is not relayed back to the brain. This has to do with too much pressure occurring in this posterior chamber, and you start to lose your visual field. The third one, is cataracts. This is actually quite common. This is when the lens, so we've talked about the lens here, starts to become quite cloudy or opaque. And actually this is pretty treatable. You can get your lens replaced in a really quick procedure. It's outpatient surgery where they make these lenses now, and the doctor just orders a new one and does a little incision in your eye and replaces that lens to fix your cataracts. And this often happens with age, that these lenses start to do that. And then finally, we have diabetic retinopathy. This is a side effect of having diabetes. The blood vessels that are in that choroid layer of the eye start to become damaged, and you will lose vision in those areas where those blood vessels have been damaged. So that's called diabetic retinopathy. All right, so at this point, I'm going to probably have you watch a video to do the dissection for the eye before we go to lab. But I am going to move on in this particular lecture and continue, and we're going to go over hearing. I'm just going to give a little bit of an introduction on hearing and equilibrium. All right, that's the introduction slide here. All right, so I like this picture because it divides the ear into what's called the external ear, the middle ear, and the inner ear. And we'll talk about the various structures as we move. So they're highlighted in blue, in sort of pink and yellow, these structures. So we'll start with the external ear, which consists of the auricle. That's the flap that kind of funnels the sound and funnels it into the what's called the external acoustic meatus, which you may remember we were talking about the external acoustic meatus and the internal acoustic meatus when we were discussing the skull. Let's look at these features in a little bit more detail. So this is the external ear. It is skin covered, right? Funnel shaped. And if you remember from our very first couple lectures, it contains elastic cartilage. And it is a supported structure, and it is called the auricle. That is the anatomical term for the ear. And this allows sound waves to be captured, all right? And it's going to send those sound waves to our inner ear. So the auricle leads to a bone, a tube, called the external acoustic meatus, which we saw when we were looking at the skull. You can see how in anatomy everything kind of comes back around again. So the external acoustic meatus, this is a canal that ends with the tympanic membrane. The tympanic membrane is often referred to as the eardrum, and this is where sound is going to transmit from. Also, deep within the external auditory meatus, the acoustic meatus, there are glands that produce a wax-like secretion called cerumen. And this is a modified sebaceous gland. And what this is doing is going to help repel water and keep it out of our ear canal. All right, so let's move on to the next segment, which is called the middle ear. We are going to talk about some of the auditory ossicles, which happen to be the smallest bones of our entire body. And they include the malleus, the incus, and the stapes. We have a great ear model in the lab too, so definitely take a look at it. And we'll also take a closer look here at the tympanic membrane. All right, so the middle ear. So sound is going to travel to the tympanic membrane, and what's going to happen is it's going to transmit sound by vibrations. So these auditory ossicles, again, which are the smallest bones of our body, they are attached to this tympanic membrane. And the sound vibrations are going to be transmitted by these little bones tapping on each other. Let's take a closer look at these ossicles. So here is a close-up of those ossicles. You can see that the malleus, it's named because it looks like a hammer. It is sitting right next to the tympanic membrane. That is the first bone that is making a connection with the tympanic membrane. The next is incus, which looks like an anvil. I actually had to look up to see what an anvil is, but it is used to hammer metal. It's like a block, a metal block. And then the last little bone right here is called stapes because it looks like a stirrup. So what happens is these ossicles are going to transmit these vibrations from sound from the tympanic membrane to what's called the oval window. So here is this oval window right here. And this is where fluids of the inner ear will move and excite receptors, sound receptors. And then those receptors are going to interact with nerves. So you can see several nerves here within the ear that are going to transmit those signals. And so what happens is the sound is going to be transformed into electrical signals, which are then sent to the brain. So I think what I want to do, though, also is highlight the terms that I want you to know, because I realize some of these slides are busy with things that I don't go over. And I also realized I circled the wrong thing here. I apologize. Here is the oval window right next to the stapes. And here are a couple of the nerves. So now we've reached this oval window. So let's talk about what happens next. And to do that, we need to talk about some of the structures that are in the inner ear. And I'm just going to focus on a couple of these structures, again, just to give you a foundation on hearing and leave it at that. But I do want to just go over what's called the vestibule and the cochlea. So the inner ear is also referred to as the bony labyrinth, and it is partitioned into 3 distinct regions. The first 2 are involved in balance and equilibrium, which is the vestibule and the semicircular canals. And then the cochlea, that is what is going to produce sound waves that initiate the action potential of the neuron to send the signals back to the brain. Let me show you what these structures look like, and then I will talk about the functions. All right, so first, let's talk about the vestibule. So here is the vestibule, and it encompasses this region right here. And to the right of it is the cochlea, that's right here. And then to the left of it is the bony labyrinth or semicircular canals. Now again, the vestibule and the semicircular canals have to do with equilibrium and balance. And the cochlea is what is going to transmit sound waves. So I want to first talk about the semicircular canals. So they are made up of a bony labyrinth. So what does that mean? So they are very intricate, delicate, bony structures that are fluid filled. So they are a bony structure, and then within this labyrinth, there are fluid filled tubes or spaces called the membranous labyrinth. And what happens is depending on how our head is positioned, there is movements of the cells that are within that membranous labyrinth. And there are hair cells that line this entire structure. And as we change our head, that fluid is going to move. And that is what is going to alert our brain that our head is in a different position. So let's look at this vestibule a little closer. So again, this whole area is going to be monitoring the position of our head. So when our head tilts, information is sent to our brain. And that is because there are these hair cells that act as receptors. So you can see these hair cells, they kind of, like this setup looks a lot like the taste buds, right? There was a receptor, and then there were nerve fibers that were associated with that. So these nerve fibers are going to transmit information depending on how this fluid flows over those cells. So if we put our head down, fluid will flow a certain way, and that will alert our brain that our head is tilted. Here is an example of how or what that looks like. So when our head is upright, you can see that the fluid is flowing. You can see these hair cells are detecting that. When the head is tilted, you get movement of that fluid, and those hair cells can actually detect that and alert our brain that our head is being tilted. Let's now switch our attention to the cochlea. So this structure, it is a hollow spiral shaped bone. It almost looks like a shell that is responsible for the sense of hearing. And it is filled with a fluid that moves in response to the vibrations that the cochlea is going to receive. There's also a nerve, and since you've now learned your cranial nerves, you will remember the vestibular cochlear nerve. This nerve innervates the semicircular canal as well as the cochlea, and it transforms the vibrations into electrical impulses, and these impulses are sent back to our brain so that we can interpret the sounds that we're hearing. So this last slide is going to show you a close-up of both those semicircular canals. The vestibule is in the middle, and then the cochlea here. So again, the semicircular canals are involved in balance and equilibrium, and the cochlea is involved in hearing. And you can see how this vestibular nerve is going to interact with both of these structures to transmit the information back to our brain. All right, so this concludes the lecture on vision and hearing and sensory receptors, and I will see you all in lab.